welcome back to me. Part 2, Chapter 9. In Part 1, we finished talking about the proton gradient. Probably sounded very odd to you. You haven't heard that before. But once again, we have a membrane. In here is the matrix. In between here, where my nose is where we're going to make a proton gradient. Out here is the cytoplasm. Okay? So this proton gradient that's going to be created, we want to build up a ton of protons in the middle here. And then on this second membrane, again, this inner membrane, we have a series of proteins that work through, that we call the electron transport chain. And at the end, we have the ATP synthase. Now, all these proteins, they get added to them with NADH or FADH2 electrons. And those electrons get passed one after the other. And as they get passed, these protons are, the energy is used to move the protons from the matrix into the intermembrane space. So they fire up and they build up to very, very high levels. And at the very end of this electron transport chain, we have this ATP synthase, which absolutely doesn't look like the scissors, but it's the best I got. And the protons will go back through into the matrix, causing that little hat to spin, as we mentioned down here, right? And making ATP. Okay, so this hydrogen gradient is energy rich in that way, not ATP specifically, but it drives the formation of ATP through something we call chemiosmosis. Remember, osmosis is a term that only applies to water, but in this case we're converting it to chemiosmosis. A chemical is doing osmosis. I don't know why they don't call it chemidiffusion or something, but in any case, this is a proton motive force, okay? We couple the energy of these hydrogen ions moving back through the membrane, through the ATP synthase, to drive and to do work. Here's all of them together. So NADH adds its electrons to the electron transport chain. The energy is used to pump protons through these proteins into the intermembrane space. You notice FADH2 adds its electrons a little further down, so it doesn't provide as much energy for ATP production. But eventually this big gradient gets built up here. Protons come back through the ATP synthase, through this process we call chemiosmosis, to make ATP. So this is how the energy flows. The majority of ATP is made by this electron transport chain. Okay, and as you can see here, once again, as sort of a big review picture, here we have glycolysis, makes 2 NADHs and 2 ATPs through substrate level phosphorylation. These 2 NADHs get moved into the mitochondrion. Now, as they get moved in, they can either remain as 2 NADHs, or they may be converted into FADH2s. If they get converted, we're going to get our lower number, 32 ATPs at the end here. If they remain, we'll get our 34 ATPs from oxidative phosphorylation. We also get 2 NADHs from moving pyruvate and converting it to acetyl-CoA. We get 6 NADHs from citric acid cycle and 2 FADHs from the citric acid cycle. Plus, remember, it makes 2 ATPs from substrate level phosphorylation. All those electrons from the electron carriers go into the electron transport chain in chemiosmosis for 32 to 34 more ATPs, for a total of 36 to 38 total ATPs from one molecule of glucose. All this came from one original glucose. 40% of the energy in a glucose molecule gets transferred to ATP. That's a lot of energy that was stored in those bonds. Now, all this, again, we call respiration. Now, because the end result, let me see if I go back, because the end result of this whole process is that these electrons that are used to power the proton gradient remain, we need to bind those electrons to oxygen, and we make water. The oxygen is the final electron acceptor. Oxygen is required for the citric acid cycle and the electron transport chain to happen. Okay? Glycolysis will still happen, but for the stuff in the mitochondria, you must have oxygen. If there's no final electron acceptor, the entire process will not work. Only glycolysis will work, okay? If oxygen is not around, instead we will do fermentation. You'll go through glycolysis, you'll make pyruvate, but then instead of converting pyruvate into acetyl-CoA and doing citric acid, electron transport, and so on, you make alcohol or lactic acid, depending on the type of organism that you are, okay? So we say that respiration is aerobic, meaning requires oxygen to produce ATP. Whereas without oxygen, we say it's anaerobic. This is a process of fermentation and, in fact, also glycolysis. So glycolysis is anaerobic. Whether there's oxygen around or not, it'll still produce ATP and some NADH. If there's no oxygen around, it's anaerobic. You'll couple this with fermentation to not actually produce ATP in this case, but to regenerate the NAD plus that was allowing glycolysis to work to begin with. And we'll show you that in a second here. Glycolysis plus reactions that generate NAD plus, which can be reused by glycolysis. Fermentation specifically itself on its own doesn't produce ATP. Glycolysis does, but fermentation allows glycolysis to keep going and going and going. Okay? If you are a yeast, you are doing alcoholic fermentation. Okay? So the pyruvate gets converted into ethanol and releases CO2. So, for example, in winemaking, which is probably not appropriate for you, but when you get to be 21, the generic process, they take a bunch of yeast, they take sugar from grapes, and they close them into big barrels, and they don't expose the yeast to any oxygen. If the yeast don't have any oxygen, they can't do respiration, they do alcoholic fermentation, and they convert all the sugar into ethanol, into alcohol. The longer you leave the sugar and the yeast there together without oxygen, the more of the sugar will get converted into alcohol. So the higher the alcoholic content, the less sugar it's going to have, kind of a balance there. You, on the other hand, do lactic acid fermentation. When you don't have enough oxygen in your cell, you can't do respiration, you'll do lactic acid fermentation, and you convert pyruvate into, you regenerate again NAD plus by making lactate or lactic acid. Lactic acid is not healthy for you, but I suppose it's better than the alcohol. Imagine if we were running a marathon, and as we get shorter and shorter on oxygen, we start producing more and more alcohol, and as we continue to run, we get more and more progressively drunk. That would probably not be ideal if our goal is to finish the marathon. Maybe if we're a frat boy, and we're 21, of course, and we want to see who can pass out the quickest, it would be ideal to do alcoholic fermentation while you're running. You'd probably see a lot of frat boys doing a lot of jogging. But that's not the case. You make lactic acid, which is actually not healthy for you, and it can build up. We won't talk about the details of that, but in any case. Here you see at the top, alcoholic fermentation. Glucose gets converted to pyruvate. Pyruvate gets converted into alcohol or ethanol. And you regenerate this NAD+, so glycolysis can happen again. This is essentially the same with humans or higher order organisms, but you produce lactate instead. The big picture point here is that you reproduce this oxidized form, the NAD+, so you can do glycolysis again with a new glucose molecule. So people talk about being, well, as you exercise further and further or more and more, you burn glucose at a faster or higher rate because your oxygen levels get progressively lower as you continue to exercise. So both fermentation and respiration use glycolysis. It's kind of that middle ground. They differ in their final electron acceptor, that is fermentation and respiration. And respiration produces many, many more ATP, on the order of 18 times more. So the main juncture, the point at which there's a split, which way are we going to go, is from pyruvate either to ethanol or to acetyl-CoA, depending on if oxygen is present or not. We definitely do love our oxygen. Glycolysis is highly conserved. It's found in even prokaryotes in their cytoplasm. And the idea is that, or it's thought that it existed before oxygen was around in the environment, which is why now it still doesn't use oxygen. Oxygen poor atmosphere during early Earth. Okay. And lastly, much of our discussion so far has been from glucose. Glucose is not the only one that goes through or allows glycolysis and cellular respiration and so on. There's all kinds of other catabolic pathways that allow it to happen. So whether you eat fats, carbohydrates, or proteins, they can all give you energy. Now this middle one, the carbohydrates, are the primary energy pathway. If you have sugars around, you're going to use sugar first. But if you don't have enough sugar, such as, for example, on the Atkins diet, there is no carbohydrate sugar breakdown. So instead, you will break down fats as your next subsequent option. Fats get broken down into glycerol and fatty acids, and they both enter the pathway at separate places to work for your ATP production. This is also why, for example, fats are nearly double the amount of calories compared to carbohydrates. Carbohydrates provide about, I think the number is 4.5 kilocalories per gram, whereas fats provide 9 kilocalories per gram. It's somewhere around those numbers. Because fats get broken up into two parts, the glycerol as well as the fatty acids that were attached to the glycerol, and they both enter this pathway. Proteins, the last option to break down protein, is not ideal, but you can break down amino acids to enter the pathway and make ATP. But one necessary evil of doing that is that ammonia gets produced, which is highly toxic to your system and can cause liver toxicity and liver damage and so on. So not an ideal option there either. Now, you can control respiration through negative feedback in the presence of allosteric enzymes within the pathway. So citrate, for example, one of the products of the citric acid cycle, if we have a lot of it, we can negatively inhibit phosphofructokinase, which was or also called PFK. It's an enzyme in the glycolysis pathway. It shuts off or turns off the system so that we don't waste and use up unnecessary glucose. ATP as well also inhibits PFK, and presence of AMP, or adenosine monophosphate, meaning all our ATP has been converted and broken down into ADP and then AMP, meaning we don't have a lot of ATP around, we need more. It turns on the system and makes it go. Okay, so ways to allosterically control and regulate the system. And on that note,